Hello guys, so today we're going to be going over and starting chapter 9. I'm going to try to make more videos about chapter 9, just breaking up the chapter into more sections. So right now we're going to be going over sections, I wrote this down, 9.1 to 9.3. So let's get into it. So this chapter is titled The DNA Structure and Analysis. So this actually, we're going to start entering like my favorite part of like history, of genetic history, like this DNA race, and this race to discover what the genetic material was, and the race to basically figure out how the genetic material worked in its structure. So it's really, I consider it quite fun. So back before 1944, everyone knew, essentially, scientists knew that genes contain genetic information. They knew that genes were passed on from parent to offspring, right? And that they were basically these heritable units, right? Starting from Mendel, which was like early 1900s. They saw that there was these heritable units that were passed on, like, from one generation to another. And they saw that this genetic information influenced the form and the characteristic of each individual and directed the complex processes that lead to the adult form. But until then, until like 1944, no one really knew what this genetic material was made out of, what this genetic information was made out of. They knew it was found on genes, the theory of chromosomal inheritance, like remember Sutton and Boveri, stated that these genes were situated on chromosomes, but no one knew what exactly the chemical composition of this genetic material was. In fact, they had boiled it down to basically two macromolecules, proteins and DNA, because they knew that chromosomes were inherently made out of protein and DNA, or nucleic acid. Because so the protein was there, as we know now today, just for the DNA to wrap around it so that it can make that compact structure feature of being a chromosome. So they knew it was between protein and DNA, and a lot of people actually thought that proteins were basically the best candidates for being the genetic information. They knew more about proteins than they did about nucleic acid. Nucleic acids, DNA specifically, hadn't really been studied very much until then. It had been discovered in when was it? Yeah, in 1868, sorry. I don't remember the date. In 1868 by Frederick Miescher, he discovered this kind of material found within like the nucleus of eukaryotes. And it was like this acidic material, and he called it nuclein. And then later on, in the late 1800s, what's the date? 1881. Okay, Albrecht Kossel who was a German scientist. He actually went ahead and like saw that within this nuclein, within this acidic substance, it was actually he went and looked at the chemical composition of it. He saw that they were like built, like he realized what the building blocks of this chemical were. And he called it deoxyribonucleic acid. So he was the guy who basically made the name DNA, right? But apart from that, there really wasn't much known about it because people didn't really consider it to be very important. Again, most people thought proteins were the way to go. There just more was known about them. They also knew that they had at least 20 amino acids that composed proteins, and so 20 building blocks that made up the protein. So this kind of showed them that there was more variety and combinations that could be made, which would basically make sense of the variety that we see in the human population or any other kind of organism that all stems from its genes, right? So there was a lot of pro-proteins. There was a lot of pro-protein candidates, and a lot of people just didn't think DNA would cut it, right? But so the genetic material, it has to exhibit 4 characteristics. That is basically replication. Yeah, it has to be able to replicate. It has to be able to store information. It has to be able to express that information and change it into something else, i.e. proteins. And it has to be mutable, right? So it has to be able to undergo mutation. And so when we look at genetic material, it has to undergo all of these things. So the first one is replication. So without replication, we basically stop the continuity of life. The continuity of life in itself stops as we know it. Because we know that through processes like mitosis or meiosis, we need to replicate our DNA prior to any kinds of these cellular divisions in order to make more cells. So this is what in the case of sexual organisms. And when we think about asexual organisms or things like bacteria, etc., they still must replicate their DNA, because in order for the bacteria to go from one cell, right, that has one DNA molecule, this is my DNA molecule inside my teeny tiny hands, okay, in order to make two progenies, both of them need to have the same DNA, because bacteria basically clone themselves. The DNA is going to encode what the bacteria is. So they have to duplicate their DNA. I know it's a different color, don't mind the different color, but imagine these being complete replicates or like clones of themselves. So two copies of DNA. And then when the bacteria divide between themselves, like from themselves, now we have two bacterial progeny, right, that have identical DNA. And this DNA is also identical to the parental or the original bacteria that we had in the first place. Then it also has to have the ability to store information. So it's basically the repository for genetic information. And this doesn't necessarily have to be expressed, which is the third point right here, expressed information. So when we have multicellular organisms like ourselves, not all of our cells are going to express every single gene found within our genome. Remember, our genome is just a collection of our DNA. And so every single one of our cells, or the majority of it, there are some exceptions, like for example, mature red blood cells don't have a nucleus, and so they don't have any DNA, essentially. So most of our cells do have the entirety of our genome, except what makes them different from each other. So what makes my liver cells different from my epithelial cells is just the type of genes that are expressed. So they all store the same amount, like they all have the genetic material that stores all the information that they need, okay? But then it's a matter of which part of the information actually gets expressed from this repository of genetic information. And this is the expression of this genetic information then gives you the actual characteristics. So this is where we go from genotype to phenotype, right? And this expression of information is pretty much like it's carried out through the central dogma of life, which is made up of two processes. So the central dogma of life states that we go from, if you can see here, we go from DNA, right, to RNA, and then from RNA, or a specific kind of RNA, to protein. And so when we go from DNA to RNA, that's known as transcription. When we go from RNA to protein, that's known as translation. Some kinds of RNA, like when we first transcribe DNA into RNA, there are some kinds that are basically stopped at that point, namely rRNA and tRNA. They don't get translated any further. They don't get translated at all, actually. They're just simply transcribed, while mRNA or messenger RNA gets translated. So essentially I'm just gonna try and stop my share and just move on to this. Okay, so the central dogma of life, again, I believe you guys know this very well. We go from DNA to messenger RNA to protein. And this is known as transcription. When we go from one nucleic acid to another nucleic acid, and then when we go from RNA to protein, it's known as translation. However, with transcription, we can also get tRNA or transfer RNA and ribosomal RNA, rRNA. And these guys don't go any further, okay? They work and they have specific roles right there, like right as they are. So this is essentially the central dogma of life, right? There's these two main processes, the transcription and translation. Right. So, and the fourth thing that the genetic material needs to have, or needs to exhibit, is the fact that it has to be a source of variation. So it has to be mutable. This is, again, important for the continuity of life. Without any kinds of mutations, there's no ability to adapt to our environment. And so this probably would cause the extinction of many species if there was no adaptation to the environment. And so again, this is just something that kind of underlies a continuity of life and how important the genetic material actually is. So the genetic material is a source of variation among organisms through process of mutation. Mutation in DNA is reflected in transcription and translation, affecting the specific protein. So once we mutate the genetic information, the genetic material, basically the DNA, through the process of the central dogma of life, through expression, transcription, and translation, that mutation will be carried through to the protein. And so the person is going to phenotypically express that adaptation, that mutation, be it a beneficial mutation or a bad, I wanted to say Maleficent. Anyways, bad mutation. And then the genetic variation provides raw material for the process of evolution. And again, evolution, continuity of life, pretty important. Okay, so right. So until 1944, observation favored protein as the genetic material. Okay, so genetic material is physically transmitted from parent to offspring. In the 1940s, proteins and nucleic acids were the major candidates for genetic material. And again, proteins were the main ones. A lot of people thought that proteins were indeed the genetic material, just because of their variety, just because of how much we knew about them, how many building blocks there were, and also because they seemed to be found everywhere in cells. Right, there was more abundance of proteins in cells compared to the number of the actual amount of DNA. This was also kind of compounded by Phoebus A. Levine, right? So another name that I would name a pug with, Phoebus, totally. I'm a pug Phoebus. Anyways, so DNA, basically Phoebus A. Levine kind of hypothesized this tetranucleotide hypothesis. So tetra meaning 4. They knew that DNA had 4 building blocks. And so what Phoebus thought was that these 4 building blocks was repeated over and over and over again. So ACTG, 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 meaning that at the end, essentially the DNA contained equal amounts of its 4 nucleotides, kind of diminishing and making that ability of making more combinations and permutations very, very small. So they just kind of underlined, or at that point, that DNA wasn't very wasn't various, right? It was just very repetitive. And so how could it at all contribute to the variety that we see around us? So the lack of chemical diversity in DNA suggested it could not store extensive genetic information, and so proteins were favored as the genetic material, right? So in came my favorite dudes, Avery, McLeod, and McCarthy, these guys right here. So in 1944, they were the first ones to kind of publish the first paper to state that DNA was actually the genetic material. And the experiment that they conducted was kind of a spin-off of the experiment that Frederick Griffith conducted back in, I don't know what year it was, when it was like early 1900s. Well, 1927, it's not that early, but anyways, in 1927, so Frederick Griffith, a British medical scientist person, he conducted this experiment that brought about this transforming principle. Okay, we actually went over this in chapter 1, but I'm going to go over it again, right? So, yeah, I'm going to stop the share and do this. Right. So Griffith's experiment in 1927, he discovered basically there was this transforming principle that transformed and changed cells, right? So he was working with a specific bacteria known as Streptococcus pneumonia. So Streptococcus pneumonia. And he noticed that there was 2 kinds of these bacteria, right? Ones without a capsule and ones with a capsule. So no capsule and ones with a capsule. The ones without a capsule were avirulent, meaning they did not cause disease. And the ones with a capsule were actually virulent, right? So because of the capsule, this capsule provided protection towards the bacteria when it got phagocytized by the immune system cells. So with the capsule, the bacteria could not get phagocytized, and therefore the bacteria cannot be destroyed. Without the capsule, the bacteria can be phagocytized and therefore can be destroyed by your immune system cells, and so you do not get the disease. But the ones that had the capsule were virulent, and therefore you did have the disease. I'm gonna do this. Hopefully that's better. Whoop! Oh my gosh. Okay, that was a bit weird. So what he did is he called the virulent strain, since it's morphologically on normal culture media, like normal ways to grow microbes and stuff. Morphologically, the virulent strain was actually smooth, so he called it the S strain, right? and the avirulent strain was actually very rough in appearance, and so he called it the R strain, the rough strain. And what he saw was when he gave the rough strain plus the little mouse, okay, that's my mouse, plus the mouse was happy, the mouse was healthy, because again, the rough strain had no capsule, the mouse was healthy. When he gave the smooth strain to the mouse died. Death. Okay, so completely unhealthy. What he did then was he took the smooth strain and he heat killed it. So he put it, like he basically essentially boiled the strain, the smooth strain. And when he heat killed it and he added it to the mouse, because the smooth strain was dead, basically the bacteria was dead, the smooth strain was dead, the mouse was actually healthy. Okay, so he took the experiment a step further. He got the R strain, right, plus a heat-killed S strain. Heat-killed S strain. So this is technically virulent, but it is dead because of the heat. And he put it into the mouse. I love how my mouse kind of looks more like a cat. I apologize, I can't draw. Okay, and when he did that, he was expecting the mouse to be healthy, because if we look, the R strain with the mouse, we get healthy. The smooth heat-killed strain with the mouse is also healthy. But when we got both of them together, the mouse actually died. So what Griffith supposed was that there was a transforming principle, there was a transforming factor that basically came from the S strain, from the dead S strain. It was something outside, basically in the environment that the S strain was in, right? And this transforming, I'm going to call it transforming principle or transforming factor, got taken up by the rough strain and changed the rough strain to make it into the S strain, right? So this was basically the first time like genetic material had been kind of seen. He was like, there's something that is transforming this R strain, this avirulent strain, into a virulent S strain. And so he was the first guy to basically see that there was this transforming factor. Okay, so he called this phenomenon transformation, suggesting that the transforming principle might be part of the capsule, or it may have been a compound that was required for capsule synthesis, although the capsule alone doesn't necessarily cause pneumonia. But so you see how the capsule was basically that important factor that caused virulency versus avirulency. And so this transforming factor either coded for the capsule or was a compound that was part of the capsule. So then McLeod, McCarthy, and Avery, McLeod, and McCarthy. I'm going to go back here and share the screen with you guys. So McLeod and McCarthy went ahead and kind of took this experiment a step further. Okay, so they wanted to actually see what this transforming principle was really, what it actually was composed of, what the chemical compound was. And so they carried out an experiment where they took this transforming factor, this transforming principle, and they subjected it to various kinds of enzymes. Okay. So they took this transforming, it's either principle or factor. I call it transforming factor just because I like it a little bit more, sorry. But it's usually principle, so let's use it, transforming principle. Okay, and they took some of it and they basically subjected it to the enzyme DNAase. Okay, so deoxyribonuclease. And so that would, by doing that, they would get rid of any DNA. Okay, so here we don't have any DNA, right, because the enzyme eats it up, but we do have RNA and we do have proteins. Okay, then they got the transforming principle again, and they gave it RNase, ribonuclease, right? So here we do still have a DNA, we no longer have RNA, and we also have protein. Okay, and then they did the same thing a third time, and this time, you probably guessed it already, they got rid of the proteins, so they subjected it to proteases. The proteases didn't touch the DNA, they didn't touch the RNA, but they did annihilate and get rid of the protein. And then they conducted the same experiment, putting it into a healthy mouse, and they tried to see if the healthy mouse died or not, right? With this this transformation principle. They noticed that when they put the ones that without the protein, right, the transformation principle still actually worked. And so the mouse, if the transformation principle works, that means the mouse dies. So the mouse died. So they were like, okay, it's not protein. Proteins are not necessary for the transformation principle to be active, right? Or yeah. And so then they looked at the transformation principle with the RNA, right? Without the RNA, sorry. So this one had DNA and protein. And so they were like, okay, what happens here? The mouse still died here in this case. That means that either DNA or protein were important for the transformation principle to work, right? So here the transformation principle didn't work, like still worked without the protein. So it means that either DNA or RNA are important. Here the transformation principle worked even without the RNA. So it's either DNA or protein. We already kind of ruled out protein from here, right? And then this third one is going to, for sure, tell us that it is actually DNA, that it makes the transformation principle so important. So here they annihilated the DNA and they only had RNA and proteins that were making the transformation principle work. And in fact, the transformation principle did not function. The mouse stayed healthy, right? It wasn't, the strains weren't transformed. The mouse remained healthy. And so they were like, okay, the transformation principle, that material that makes it up is actually DNA. And you can imagine just kind of like how, it wasn't something that a lot of people agreed with, and so a lot more experiments had to be done in order to actually prove that DNA was indeed the genetic material. So then we move on to the second major evidence, because again, there were so many people who were so steadfast in thinking that proteins were actually the genetic material, that it wasn't, that first wonderful experiment by Avery, McLaurin, McCarthy wasn't very well received, right? So the second major evidence came from Hershey and Chase. So we had Alfred Hershey and Martha Chase in 1952. They helped basically clarify the just what the genetic material was. And after this experiment, there was no doubt basically that DNA was the genetic material. So what they ended up using were bacteriophages. And we kind of talked about bacteriophages in chapter 8, but we know that bacteriophages are viruses that specifically infect, right, bacteria. And so to kind of look like this, they used a specific virus known as the T2 virus, right? And what they knew about the T2 virus was that it was 50% protein and 50% DNA. So again, we could understand, was it the protein or is it the DNA? Is it the DNA or is it the protein that allows for the bacteriophage to basically force its host to make more of itself, right? To force its host to make more viruses, more bacteriophages. The other thing that they knew is that the infection started out by when the tail fibers actually absorbed or attached to the bacteria. So this right here is my bacteria. So when the tail fibers attached to the bacteria, that's when we had infection. And basically, they did it because once this absorption happened, the phage was able to produce more viruses within the bacteria cell by essentially hijacking the bacteria cell. Okay, so the phage would insert its genetic material inside the bacteria, whatever that genetic material it may be it protein or DNA, right? And then that genetic material would then be replicated and expressed, right, into making more phage particles, which would then assemble and make more viruses. Okay. So it appeared that basically some molecular component entered the bacteria cell and directed bioreproduction. So what was this molecular component? So what they ended up doing was they took a virus and like the same virus, but two different test tubes of it, and they labeled it with using radioisotopes. They labeled one with 32P, the radioisotope 32P. And this 32P labeled the DNA, right? And so here we have our DNA that's nice and labeled. This is because in DNA we do have phosphor, and so our DNA, any place that we had phosphor in our DNA had this 32P. Then they took the other one, let's take another color, the other batch of viruses, and they labeled it with 35S, right? This 35S was only found in proteins, because proteins have sulfur. DNA does not have sulfur in any of its, in its actual molecular composition. It doesn't have any sulfur. Okay. And then, so they had these labeled DNAs, and they went ahead and they allowed these, labeled DNA, sorry, labeled phages, and they allowed these phages to infect the bacteria. So they infected the bacteria, okay? And what they ended up seeing was that with the protein, most of the protein stayed outside. So it was like basically because this phage was green all over, right? The protein stuff stayed outside. It was kind of like this empty ghost shell, this empty spaceship, and then something else was put inside. But inside the bacteria, we didn't have any 35S protein. What they did see with the DNA, however, with the 32P labeled phage, was that we had the phage itself stayed outside, right? The structure of the phage itself stayed outside, but what was inserted was this genetic material, and the genetic material was labeled with the 32P DNA. In addition, what they saw was that after viral replication happened and lysis and rupturing of the bacteria occurred, we got more little phages that had this 32P DNA. So basically the progeny of the progeny of the viruses, the viral progenies, continue to have this genetic material. You see what I'm talking about? So they continue to have the 32P DNA. And so with this experiment, basically Hershey and Chase put to rest the fact that proteins were the genetic material. In fact, they said that they definitely were not, right? And they really basically proved the case that DNA was the genetic material.